Okay, welcome to this explainer. Today, we are diving straight into the high-mark exam strategy for eukaryotic cell organelles. So let's kick things off with a pretty harsh reality. Most students end up throwing away easy marks on this topic, but stick with me, because by the end of this, you definitely won't be one of them. Take a really close look at this cold open mistake. It's a super common, totally generic student answer that honestly seems completely fine on the surface. Mitochondria produce energy for the cell to use. I mean, if you've been revising this topic, you've probably written something exactly like this on a flashcard, right? It sounds like that solid foundational biology stuff we all learned years ago. But here's the catch. Now, what's really wild about this is that it exposes the one tiny mistake that costs candidates an entire grade boundary. Seriously, why does this generic answer score absolutely 0 marks? Nothing. Nada. Well, it's because at the AS and A level, the whole theoretical architecture of biology rests on really precise definitions and causal relationships. You see, energy cannot be produced. It's released or transferred via ATP. But honestly, even beyond the scientific freezing, there's a much bigger structural failure happening in that answer, and it's one that examiners see all the time.
All right, moving into section 1, the costly organelle mistake.
Here's the deal. When you just memorize facts about cell organelles totally out of context, you're relying on superficial breadth. But examiners, they're hunting for analytical depth. This comparison here beautifully illustrates the difference. The flawed side is totally generic, while the top band side takes specific organelle terminology and applies it directly to the context of the question. Instead of just vaguely saying mitochondria do something with energy, the top band candidate links the organelle's physical structure to its actual function within a specific scenario. Like, if the question is about a muscle cell, the top band answer explains how the high density of mitochondria provides ATP specifically for muscle contraction in that exact moment. You absolutely have to integrate your knowledge with the specific syllabus area they're testing.
Let's shift gears to section 2, unlocking the method mark strategy.
So, the crucial takeaway here is that examiners don't just want a list of facts. They actually award marks for your analytical process. You aren't just some biological encyclopedia spitting out data. You need a framework. In just a second, I'm going to show you the ultimate examiner clue that will literally change how you structure your answers. But first, you need the procedural framework that makes that clue perfectly obvious. Because without it, you're going to keep dropping those easy points. Not because you don't know the biology, but because you aren't communicating it in the specific language the mark scheme is begging for.
Which brings us to section 3, the answer framework.
You're going to want to lock this path into your memory. Step 1, deconstruct what they are actually asking you. Step 2, extract the relevant data from the stimulus materials. And please, whatever you do, don't ignore the graphs or texts they give you. Step 3, select the analytical principles you need to apply. Step 4, execute your analysis, showing clear working. And finally, Step 5, verify your results and present them using precise, correct terminology. That is how you secure top bands. Systematic processing every single time. Okay, on to section 4, analyzing data and statistics. Now, biology isn't just about drawing pretty diagrams of Golgi bodies. It involves some heavy, rigorous data handling. When you're faced with experimental results about organelle function, you have to know how to process those numbers mathematically. If you ever freeze up wondering which test to use, just think back to this. You have to perfectly align your data type with the correct analysis. Use a t-test when you're comparing the means of two different sets of data, like say the mean number of chloroplasts in two different leaf environments. Use chi-squared for categorical data, evaluating expected versus observed outcomes. And grab the correlation test when you're looking for a relationship between two continuous variables. Definitely mentally bookmark this table. It's actually amazing to see how top-scoring candidates constantly demonstrate this evaluative thinking when they look at cell biology evidence. Rather than just describing what they're looking at, they evaluate alternatives, they compare different approaches, and they justify their conclusions with hard evidence. When you are looking at a comparative analysis of organelles, it requires pointing out similarities and differences, using proper comparative connectives, and drawing smart conclusions about why those differences actually matter. Let's move on to section 5, Principal Examiner Top Warnings. We're cracking open the Cambridge Principal Examiner reports here to look at the exact traps you absolutely need to avoid. These are the top 3 mistakes candidates make, and they are directly responsible for bleeding points on what should be straightforward cell structure questions. First up, confusing cause and effect. Did a missing organelle cause the cellular defect, or did an external factor cause the organelle to fail? Don't mix those up. Second, forgetting units and proper terminology. Just saying a cell is bigger is totally useless. You need specific micrometers and proper descriptive terms. And third, providing answers that completely lack specific context. And that brings us to the vital examiner clue I promised you earlier. Context is literally everything. Failing to link your knowledge to the specific scenario they gave you is a fatal error. This is exactly why our cold open mistake from the beginning scored a 0. The candidate wrote a true biological fact, yeah, but it was just floating in a void. It wasn't linked to the scenario in the prompt. If the question gives you a context, a specific disease, a unique plant cell, specialized mammalian tissue, every single point you make must anchor right back to that exact context, no exceptions.
So, to really lock this in, use this simple memory hack, taking the first letters of our framework. It will basically guarantee you never miss an analytical step in the exam hall. DESEP. Deconstruct, extract, select, execute, present. When you sit down and look at a huge, complex, multi-part question on organelles, just quickly write DESEP at the top of the page. It forces you to pause, analyze the stimulus data, choose your framework, and present a highly contextualized answer. It's a real game changer. And hey, never forget this magic number. You are being assessed on exactly 3 cognitive domains: knowledge, handling information, and experimental skills. It isn't just about what you know. It's about how you handle the completely novel information they throw at you and how well you understand the experimental procedures used to study these cells. If your revision only focuses on domain 1, just memorizing knowledge, you are severely capping your maximum possible grade.
All right, let's jump into Section 6, the Diagnostic Exam Challenge. Let's put your new framework to the test right now with a high-stakes interactive diagnostic challenge. Pause for a second and predict your answer before we reveal how the examiners would correct it. Think about the specific command word here, evaluate. You are looking at data regarding an organelle defect. Based on the framework for higher order assessment we just talked about, what 3 critical elements absolutely must be in your analysis to get full marks? Okay, let's see how you did. If you hit all 3 of these points, you are executing the critical analysis exactly the way the mark scheme demands. Evaluating evidence quality means you have to state the limitations of the data, point out any assumptions that were made during the experiment, and offer alternative interpretations of those results. You also need to explicitly state how those factors affect the final conclusions. This is what true, top-band evaluative thinking looks like on paper. But wait, think about this for a second. What if the command word was just describe instead of evaluate? Your entire high-mark strategy completely collapses if you misread that one word. A beautiful, well-thought-out evaluation scores 0 if the examiner only asked you to describe what you see. Crazy, right? So how much of your grade is currently at risk simply because of the command words you might be skimming over? Keep checking out our explainers to excel in your Cambridge examinations. Keep practicing that DESTEP framework and focus on the context. I'll see you in the next deep dive.